Mission Gesundheit. Am Puls des medizinischen Fortschritts mit Dr. Hadi Saleh.
Herzlich willkommen zur 8. Folge Mission Gesundheit. Ich bin Dr. Hadi Saleh. Ich bin ein großer Fan von Wearables. Ich trage selbst am Körper ein Whoop-Armband, um meine Belastung, meine Erholung und meinen Schlaf zu messen, eine Apple Watch für mein Training und zeitweise ein Glukosemessgerät, um meinen Blutzucker zu messen. So sehe ich immer genau, welche Lebensmittel, welche Sportart und wie Schlaf sich auf meinen Körper auswirken. Und dabei habe ich etwas beobachtet und einen Trick für Sie.
Hadis Health Hacks.
Vor dem Essen Essig in ein Glas Wasser geben und trinken. Essig, insbesondere Apfelessig, wird oft zugeschrieben, dass er die Verdauung unterstützt. Die Säure im Essig könnte helfen, den pH-Wert im Magen zu senken, was die Verdauungsenzyme effizienter arbeiten lässt. Einige Studien deuten darauf hin, dass Essig das Sättigungsgefühl steigern und den Appetit verringern kann, was beim Abnehmen helfen könnte. Vor allem aber kann Essig die Insulinempfindlichkeit verbessern und die Blutzuckerspiegel nach dem Essen stabilisieren. Dies ist besonders für Menschen mit Diabetes oder Prädiabetes von Vorteil. Essig enthält Antioxidantien und andere bioaktive Verbindungen. Das fördert ein gutes Hautbild, unsere Augen und kann den Körper vor Krebs schützen. Zum Beispiel regt Essigsäure die Produktion von Speichel, Magensäure und Verdauungssäften an. Hier aber immer auf die Dosis achten, denn Essig ist sauer und bei übermäßigem Gebrauch kann es den Zahnschmelz schädigen oder Magenbeschwerden verursachen. Ich bekomme regelmäßig komische Blicke im Restaurant, wenn ich mir gleich zu Anfang Essig bestelle, ist aber wirklich sehr effektiv. Als Kind wollte ich immer Cowboy werden, aber später als Jugendlicher ist mir klar geworden, dass ich am liebsten Menschen helfen möchte. Deshalb bin ich Arzt geworden. Die beliebtesten Berufsträume von Kindern variieren oft je nach Kultur und Zeitgeist, aber einige Berufe bleiben durchgehend populär. Zu den häufigsten Berufsträumen von Kindern gehören Feuerwehrmann, Tierarzt, Arzt oder Krankenschwester, Polizist, Sängerin oder Profisportler. Aber kaum ein Berufsfeld löst so viel Faszination aus wie die Raumfahrt. Was das mit unserem nächsten Gast zu tun hat und wieso er im Grunde gleich 2 Kindertraumberufe auf einmal ausübt, hören Sie jetzt.
Talk der Woche.
Mein Experte in dieser Woche ist Christopher Mason. Dr. Christopher Mason ist Professor für Genomik, Physiologie und Biophysik bei Weill Cornell Medicine in New York. Er erwarb einen Doppelabschluss in Genetik und Biochemie an der Universität of Wisconsin-Madison, einen Doktortitel in Genetik an der Yale University, ein Postdoktorandenstipendium in klinischer Genetik an der Yale Medical School und er war gleichzeitig der erste Gastwissenschaftler für Genomik, Ethik und Recht am Information Society Project der Yale Law School. Ein Mann mit vielen Abschlüssen also. Chapeau. Er ist Mitglied in zahlreichen medizinischen Einrichtungen und Organisationen, darunter dem Konsortium für Weltraumgenetik der Harvard Medical School und des New York Genome Center. Das heutige Interview deswegen auf Englisch.
Experte der Woche.
Thank you very much for joining us for this episode and great to have you.
Yeah, a pleasure to be here.
So your lab develops and utilizes biochemical and computational technologies to understand human genomes. Could you explain it to me like to a 5-year-old?
Yes. Well, basically, when you look at your hands and look at your body, of course, there's every cell carries a bit of your genetic information, or I should say almost every cell has a bit of the DNA. And in that DNA is your risk for disease, what might keep you healthy, how you respond to a drug, basically how you respond to the environment. And so that genetic code is also part of the reason why you have muscle cells and you have neurons and you have different skin cells. The genetic code is the same in your whole body, but the way it's used is different, as evidenced by the fact that we have different organs in our body, right? So we have different heart tissues, different muscles, cells, different cells do different functions in your body, and they also respond differently. So we try to understand literally almost every cell in the body and how it changes and responds to stressors, to disease, including even to space flights. We want to try and understand the human body and also microbial cells that are also hanging out with the human body. And then once we understand them better, even modify them or even change them so they can, cure disease in some cases or treat against really big stressors like going into space.
So can you give us a couple of examples, how your findings can help treating diseases?
Yeah, so some of them have looked at epigenetic differences, so how genes are changed. So for example, in leukemia, we see what's something that's called a methylation, where essentially it's like when you decorate, say, a Christmas tree with ornaments, you get, essentially little colors on them. The same thing can happen to your DNA, where it's decorated, it is changed, it's modified based on these epigenetic marks, which if cells are behaving normally, they work great and it looks like a normal Christmas tree. But if it looks like someone randomly, essentially threw a bunch of garbage on your tree and took a bunch of branches, as a metaphor, that would be what happens sometimes in cancer cells. And so we found that this change of regulation can sometimes lead to aggressive cancers. But more recently, it's also places that we can target it. So we can reprogram these changes and put them back to the way they're supposed to be, as a way to cure potentially leukemia or lymphomas. Or more recently, actually, there was a clinical trial that was just approved for now broad treatment for sickle cell disease and beta thalassemia. Basically, you turn back on fetal hemoglobin in your body if your adult hemoglobin, which carries oxygen in your body, is basically has a defect.
So in Europe, there are 14 different gene therapies approved, and I know there are many in the pipeline. Can you explain to us what happens during a gene therapy?
Yeah, there's different kinds of gene therapies. So some of them really have a kind of like a little mini package, almost like a little delivery that's going into the house. And if your house is your DNA and your genome, it might land in your kitchen counter, it might land in your bedroom, but it carries with it kind of like a signal or essentially really a full package of genetic payload that either carries a corrected version of a gene that has a defect, or sometimes even a new therapy that could modify your T cells. That makes like maybe in your house, again, using your house as the metaphor here, you have your bedroom, but if suddenly you had someone trying to break into your bedroom, if you somehow could combine a metal door with a big lock that are in two different rooms, you can make a chimeric version or a combined version. And that's actually what we can do with T cells. The immune cells that attack cancer, you can reprogram them to make them essentially a new version of a cell that targets like new components of the house of your body put together that can target specifically the cancer surface markers. And so these are two examples where you either turn on a gene, or you can actually bring a genetic payload or even rearrange parts of your DNA and add new DNA to give it a new function. So they come in many flavors, but the ultimate goal is to use genetic engineering to cure a disease.
So let's digress for a second into space. Is it true that you wanted to become an astronaut?
Well, I think if someday we'll all become astronauts. I don't think I want to be. I think I know at some point it'll be routine for us to go to and from the space station to the moon. Yes, I would like to, but I'm in no rush. I want to make it so there's very comfortable seats, and there's maybe some cocktails on the moon or something very relaxed. So that would be the ideal. But I would even go if it was a more dangerous mission, and if it was even a one-way trip. If I'm, say, I don't know, in my 75, 80, and I need people to go for the next 10 to 20 years and say, listen, it's a one-way trip, and figure, well, you gotta die at some point. You might as well do it on the frontier. So I'd be happy then. But I'd want to make sure our daughter's older and it's less tragic if I die when I'm older, I'd say. So something along those lines.
Have you applied for Astronaut Academy at NASA?
No, not yet. No, so same reasons. I think I wouldn't want to, even if I got in, I wouldn't leave anytime soon. So for family purposes, but maybe in five years or so I could.
So let's talk about the connection with NASA and your work. With the what's it called, the Consortium for Space Genetics. So what is it that you do there?
So the consortium is a group of researchers mostly from Harvard, also some from Cornell, and collaborators, the people who work at NASA or the Japanese Space Agency, or more recently we've been working a lot with SpaceX for a variety of missions. So the consortium is to really think about space flight from the genetic perspective and think, okay, what do we need to survive in space? What genetics we carry, but also the microbes that we bring with us. How could they help us survive in space or help us create food on the moon or on Mars? Because we'll need, of course, to eat. We'll need sustenance. And the way we do it on Earth, of course, really heavily relies on our microbial friends. And we'll need them on the moon and Mars. But they've never lived there, or as far as we know, they've never lived there. So we'll probably have to help them, nudge them, maybe direct their evolution so that they can more functionally survive and create food and nutrients for us on these new moons and new planets. And so the consortium is everything from human biology to microbial biology to computational methods to thinking about genetics and how that changes in space.
There is this famous twin study that you contributed to. Can you elaborate on that study and the two astronauts that you examined?
Yes, we had a really great, I was very honored to be selected by NASA to be one of the principal investigators for that mission, which was the really simple idea of we're identical twins. Let's put one into space for a year and keep one on Earth and then see everything that changes. So getting blood samples from space, again, microbial samples, doing skin swabs, doing lipid profiles. We're looking at fat molecules. We looked at cognitive metrics, looked at eyes, the vasculature, really the full armamentarium of molecular biology and physiology we brought to bear to understand really at the highest density ever what happened to the body in space. And we found that, it was really extraordinarily plastic, that many things that change, almost all of them reverted back to normal within a few weeks or months of getting back on Earth. There was some big surprises, like his telomeres, which is the part of the end of your chromosomes that keep your DNA intact, which, normally they shrink as you age because they kind of slowly degrade, kind of like a carpet slowly degrades. But his actually got longer in space. So that was a really surprising result. And it did go away, though, when he came back to Earth. So the fountain of youth is not forever in space, and he did get a lot more radiation. So it's not necessarily the best place to hang out forever. But, a lot of what we publish and have said since for other missions is that it seems like space, while very stressful on the body, is not really insurmountable. And if anything, your body adapts quite quickly. You have what are called chicken legs in the first few days, where all the fluid moves to your upper body. But then within a few days, it regulates and you start to look normal again. So this is something that the body, while very confused when it's in space at first, has a really extraordinary ability to respond and adapt. And even if it's being radiated and having different food and microbial and immune stressors, all these things happening, it still does a really extraordinary job to get back to normal in space and when it gets back to Earth.
Have you looked at changes in methylation patterns of DNA, and was there any difference?
Yeah, so getting back to kind of the ornaments on the Christmas tree idea of the methylation marks, we did look at DNA methylation for Scott versus Mark, the kind of the two twins, and we could see a lot of the changes in the immune activation. So you could see his T cells really actively, becoming aware of the new environment and looking essentially for new targets, these immune cells that help keep your body protected. And, so we could see that there was a sense of a lot of activation of genes as well. We could see that the body, flipping on a lot of light switches to react to the stress of space. And she said there was, thousands and thousands of these genes that were activated. And we could also see the epigenetic change adjacent to those genes that were changing. So it does indicate there's a broad response that we can see. And we've now seen this for other astronauts as well. We see telomere elongation. We see epigenetic changes. We're doing some follow-up studies now on epigenetics as well.
Are there any results that we can use for, treatments here on Earth?
Yeah, a couple of things that that have been discoveries that are going to be really helpful for on Earth, and also some of the technologies helpful on Earth. So one of the biggest applications, I think, of some of the knowledge so far gained is a lot of the markers for aging actually are accelerated in space. So osteoporosis, or muscle loss, or even some of these epigenetic marks of aging seem to be perturbed. So we think spaceflight can help us better understand diseases of aging because it accelerates almost all those same features. And the other thing, that's interesting is also we see some of these same changes for infection response. So, some of the markers when we look at the immune activation in spaceflight looks a lot like when you have an infection on Earth. And so it actually shows as a model for how does the immune system respond to stress and how does it actually kind of counteract it. So it gives us another way to study those questions. And then what's really exciting is also getting new technologies in space. So actually we got a little miniature DNA sequencer working in space in 2017 and 18, which is now used in routine missions. And so even the ability to do like a diagnostic, like if you have an infection in the middle of the rainforest and you want to know what it is, we could grab a sample, extract it, sequence it, and tell you what is happening. And also we can sequence blood in space now. So we can actually take blood from astronauts in the middle of the mission and get a really quick profile. So the miniaturization of spaceflight for technology forces you to be very nimble, use very few supplies, and do it with as few steps as possible, because you have an astronaut doing it that doesn't necessarily have a medical background, although usually some of them do. And so this has forced us to be very, I think, again, nimble and really collaborative with technologies to get them so they work in space for diagnostics.
And obviously they have overcome working in zero gravity, right? Yeah.
Things float around. There's also, things float as well as little bits of microbial matter maybe left over from the bathroom and the toilet. There's, food. There's little bits of droplets. So it's a funny environment, of course, because the space station gets almost 99% of its water is reclaimed. So it does a really good job of capturing all the water, just say from humidity, from sweat, from urine, for any waste. And I remember Scott Kelly, when he got back, he was being interviewed, and he said, someone, is it true that you drank your own urine effectively because it was recycled? And he said, well, yes, but not just mine. It was everybody's up there. So it all gets recycled, but it's an extraordinary feat of engineering and technology to enable purification, cleaning, and you can use that water right away again. So it really is, these great technologies that, again, can help us. How do we purify water in the middle of, say, sub-Saharan Africa, or if we're in the middle of remote parts of Asia, or even in the United States, a remote area where there's a place there's just not that much water. Any bit of water you can reclaim and capture can help, people here on Earth, and eventually people that might go to the moon or Mars.
That's great. So your research lab has now released, a couple of open source software packages for genomics, epigenomics, metagenomics, and machine learning. And in the spirit of making it available to the broader public, has it all played out the way you planned for it?
Yeah, if anything, yes, I'd say the short answer is yes. I really am glad when we release algorithms open source for the community to use. Some of them have been also commercialized and used by companies to get out to the market. But for the most part, the code starts to be as open source and open for anyone to use, because I think it is a really simple way that people can build upon prior work much faster. So, if you have to negotiate a license or it's secret for a while, it's harder to build upon it. Whereas, not impossible, like some of the ChatGPT tools or OpenAI is part nonprofit and part for-profit. So there's ways you can kind of do both. But whenever possible, we try to have the code open, and it's now been used. Actually, one of our most highly cited papers was just a software package, but it's one that was so useful for epigenetics. It's one that thousands and thousands of people have been using for their research. So it's very gratifying and kind of humbling to know that something we put together, over a couple weeks and some coffee and thinking about what would be, mostly like what would make our lives easier, like what's some algorithm we can write that just makes the code easier and the model more useful, and make our science more applicable, that other people found it useful as well. Because I've written many pieces of code and software over the years, and some of it just sits on a small GitHub page, and 5 people use it or something. But it's nice when a lot of them get used by many people.
So let's take a brief look into the future. How do you foresee the future in the next 10, 20, or 30 years? Will we be able to cure cancer? Will gene therapies will be widely available? How do you foresee the future?
I've thought about this a fair amount because I just wrote a book called The Next 500 Years. So I think 30 years is, in theory, not too hard. But I'd say the next 10, 20 years, we still have a lot of fundamental discovery that has to be done about the human genome, about the microbiome, all those microbes like bacteria and viruses that are with us and on us and in us. And so we, are still even discovering human genes. Like, you think of how many genes does the human being have? Is it 10,000, 20,000? The answer is about 60,000 these days. But that's 10,000 more than we had 10 years ago, right? So we're still discovering the fundamental building blocks of the human genetic code today as we speak. It's not completed yet. Even though the human genome, the draft genetic code, was published in 2001, we're still teasing out where are all the true components. So I think some of that work will still continue for the next 10, 20 years, especially in the microbiome. But that, this ability to customize gene therapy for cancer patients is now in the thousands of clinical trials globally. Doing CRISPR therapies, it's now approved in the United States and the UK for beta thalassemia and sickle cell disease. Is extraordinary. It's amazing, and it's just some of the very first work that could apply to probably almost every disease, where you could think about either genetically or epigenetically modifying specific sites to be curative, which is extraordinary. But it will take, CRISPR was first discovered in the late 90s. It took a while for it to be characterized. It took time for it to be deployed, perfected for use in human cells. But what's happening today, there's already been what are called, some of these omega proteins and other tools that Feng Zhang and others have published, where we have entirely new genome editing systems that are being discovered as we speak. And so it's presumed that over the next 10, 20 years, they'll also get better. We'll find the ones that are more specific or safer. And, so 20, 30 years from now, we'll probably have really a good, like a whole tool belt of genome and epigenome editors that we could bring to bear for any disease, or maybe we could look at aging differently. Maybe we could try and tweak some of the aging cells in our body and try and maybe push that back a little bit as well. So I think we'll see a lot more tools, and then start to be developed for people that aren't yet sick but want to avoid getting sick, I'd say.
Yeah, for every 4 years that we live, we get one additional year. And people like George Church or Ray Kurzweil, they speak of the longevity escape velocity. And they, foresee it to be happening, let's say, in the next 15 to 20 years. And when that happens, for every year that we live, we get one additional year.
Yeah.
How do you think about that?
This idea that if you can just live a little bit longer, you'll actually maybe get to live to be 120, 150, 200. I think that is likely. You'll definitely live longer on average than I think anyone has in history. I mean, writ large, life expectancy keeps going up, except for COVID, it dipped down. For the global influenza in 1919, it dipped down. For global world wars, it of course dropped down the global average. But it's continued to march up. This is the average life expectancy now in the mid 70s for many industrialized nations will probably keep going up. But I think what's interesting is the escape velocity will require a bit more intervention to really become more than just moving the average up. The maximum lifespan has not fundamentally changed in 100 years. It's really about 120. There's one person maybe lived to be 126. And that's been the same for people who just smoked and drank every day, allegedly. So maybe the trick is to drink and smoke every day. Of course, I'm joking. That's almost certainly not the case. But it just goes to show the later edges of lifespan we scarcely understand, and people don't live that much past 105, 110. It barely ever happens again. So I think there's this maximum end of lifespan that we may only be able to push back if we think about some of these genome and epigenome editing tools, or substantive changes in lifestyle and nutrition that might border on essentially drug regimens because they'd be so engaged. But I think about the end. If you look at life expectancy plots, the average moves up, but the maximum has not fundamentally changed in probably 100 years. So, and if anything, that was a few decades ago that the maximum lifespan was reached. Which to me means there's some limit that's out there at the end that we might have to get more inventive if we want to push through. I've met people who say, I'm going to live to be 200 years old or 300, or I've even met people who say, I don't think I'm ever going to die. To which I say, I hope you're right. I think that's great, but you should plan just on the outside chance that you might. That's probably not going to happen. In the history of all things that have ever lived, they've all died. But I wish you the best of luck, and I'm happy to help any way I can. Is basically what I say to people who think they're never going to die. But that's what we do need. People that want to push the envelope to learn more about some of these questions of how long can cells live, how long can you use one organ before you have to replace it, for example. Can you use, if we can start to get xenotransplantation working, for example, where you have organs from other animals, maybe you can start to swap out organs, and that'll give you another 5-10 years too. So I think a lot of these ideas that were kind of very early in the 60s and 70s were, the very first transplants happening then from xenotransplants or bone marrow transplants of modified bone marrow. So, they're really only a few decades old, some of these really significant interventions in health and organs, basically. So I think if you look at it on a long enough timeline, there's actually a decent chance we can push back that maximum.
Do you have a specific age number in mind for yourself?
Yeah, my current plan, or I guess what I'm banking on in terms of when I do vacations, what I think about retirement savings, I think I could make it to 100, but I'm not planning probably past that just life-wise. I think if anything really past, I'd say 85, 90 is a bonus. But my grandparents lived to be in mid-90s. I think, I'm relatively healthy. I think 100 is a reasonable number to get to. But also the other thing is that, as we all talk about, you don't just want to have lifespan, you want to have health span. You want to be able to live healthy. So if I'm, 102 but can still move around and, wrestle with my grandchildren, then I'll be really happy. But if I'm just a big blob in a chair and I can't move, it'd be less exciting, I'd say, yeah.
Yeah, nobody wants that.
Yeah. So I think, but it's, I think when I was younger, when I was a kid, I actually remember recently was looking up the orbital trajectories for Halley's Comet, which when it returns back to Earth, I'll be 86. And I remember when I was a kid, it was in 1985 that it came through. I was only 7 years old, and I missed it because I was, a kid, so I didn't know to go see it, or we just didn't have a family event for it. We were busy. And I remember when I was a kid thinking I probably won't live to see when Halley's Comet comes back. But now I probably will. So it's even within my own lifespan, I think my optimism towards a longer life has gotten better.
That's great. So Chris, thank you very much for joining us today. It was a very interesting and inspiring discussion that we had. And on that positive note, Halley's Comet,
yeah,
we all get to live to observe it. Thank you very much for joining us.
It's a pleasure. So thanks so much for having me.
Die Fakten der Folge.
Chris hat erklärt, wie genetische Informationen in jeder Zelle des menschlichen Körpers verschiedene Faktoren wie Krankheitsrisiko, Gesundheitszustand und Umweltreaktion bestimmen. Diese Informationen variieren in ihrer Nutzung zwischen verschiedenen Zellen und beeinflussen deren Funktionen. Jede Zelle im menschlichen Körper trägt genetische Informationen, die für die Krankheitsrisiken, Gesundheit, Medikamentenreaktionen und die Reaktion auf Umweltfaktoren verantwortlich sind. Es gibt verschiedene Arten von Gentherapien. Einige Ansätze umfassen das Einbringen einer korrigierten Genvariante oder das Modifizieren von Immunzellen wie T-Zellen zur Bekämpfung von Krebs. Diese modifizierten T-Zellen können gezielt Krebszellen angreifen. Die Behandlung von Leukämie erfolgt beispielsweise durch das Verändern von epigenetischen Markern oder die Nutzung von Gentherapie für die Behandlung von Sichelzellanämie und Beta-Thalassämie. Forschung befasst sich damit, wie Genetik das menschliche Überleben im Weltraum unterstützen kann und wie Mikroben das Leben auf anderen Himmelskörpern ermöglichen können. Dafür ist die Anpassung mikrobiellen Lebens an außerirdische Umgebungen für die Produktion von Nahrung und Nährstoffen notwendig. Die Studie, in der einer von 2 eineiigen Zwillingen ein Jahr im Weltraum verbrachte, half die Veränderungen im menschlichen Körper durch die Raumfahrt zu verstehen und enthüllte, dass diese zwar signifikant, aber größtenteils reversibel sind. Wir können also optimistisch in Bezug auf zukünftige Durchbrüche in der genetischen Medizin sein und blicken maßgeschneiderten Gentherapien für verschiedene Krankheiten und möglicherweise eine Verlängerung der menschlichen Lebensspanne entgegen. Das war eine weitere Folge von Mission Gesundheit. Vielen Dank, dass Sie auch heute wieder Teil meiner spannenden Reise durch die Welt der Medizin geworden sind. Ich hoffe, dass Sie wertvolle Einblicke und Inspirationen aus unserer heutigen Diskussion mitnehmen konnten. Denken Sie daran, dass Gesundheit das wertvollste Gut ist, das wir besitzen und dass jeder Schritt, den wir tun, um unser Wissen darüber zu erweitern, ein Schritt in Richtung eines besseren Lebens ist. Abonnieren Sie uns gerne, um auch in Zukunft keine Episode zu verpassen.
Hardys Random Reminder.
Proteine, Proteine, Proteine. Denn Proteine sind essentiell für den Aufbau und die Erhaltung von Muskelmasse. Sie sind besonders wichtig für Menschen, die regelmäßig Sport treiben, sowie für ältere Menschen, um Muskelabbau zu verhindern. Viele Enzyme und Hormone, die in verschiedenen Körperprozessen eine Rolle spielen, bestehen aus Proteinen. Auch viele Antikörper, die helfen, Krankheitserreger zu bekämpfen, sind aus Proteinen zusammengesetzt. Und sie spielen eine Rolle beim Transport und der Speicherung wichtiger Nährstoffe im Körper. Also mein Random Reminder diese Woche: genügend Proteine essen. Am besten ca. 1,5 bis 2 Gramm pro Kilogramm Körpergewicht. Das heißt, bei einem Körpergewicht von ca. 80 Kilogramm 160 Gramm Protein essen. Besonders proteinreiche Nahrungsquellen sind Hülsenfrüchte, Hüttenkäse oder auch jede Form von Fleisch. Und selbstverständlich Eier. Bis zum nächsten Mal. Bleiben Sie gesund und neugierig.
Das war Mission Gesundheit. Am Puls des medizinischen Fortschritts mit Dr. Hadi Saleh. Eine Produktion von Ceramtech, einer der weltweit führenden Medizintechnik-Plattformen. Sie haben eine Frage an Dr. Hadi Saleh? Schreiben Sie uns an podcast@ceramtech.de.